So that's two milestones. The first milestone is the one I just mentioned. It's the largest percent increase we've ever seen in the ITP. The other milestone is the first time we've ever seen a pair of drugs that used together gives you a greater percentage increase than either drug used by itself, and to a significant extent.
So hello and welcome to the Sheekey Science Show, where in this video I spoke with Richard Miller, Professor of Pathology at the University of Michigan and the Director of Michigan's Paul F. Glenn Center for Biology of Aging Research. He's also a lead driver behind the Interventions Testing Program, where they study the effect of various drugs on lifespan in mice. Here we discuss further the ITP study, as well as his lab's research into understanding the mechanism of aging. I hope you enjoy our conversation. So, hi Rich, thank you for joining me today and welcome to the Sheekey Science Show.
I'm pleased to be here. Thank you for inviting me.
Oh, thank you. So I thought before we go into some of your recent research, it'd be very interesting for my audience to hear a bit about maybe your introduction into science.
That is, in my personal reasons for getting into science, is that the question?
For sure, yeah, that, and also maybe touch upon some of your earlier research. I know you did some work on the immune system.
Sure, yeah. So when I was, 10 or 11 or 12 years old, it occurred to me that getting old was a really rotten idea. People tend to get sick and then they died, and none of that sounded like fun. So it occurred to me the smart thing to do would be to learn how to slow the aging process so that people, including myself most importantly, could live a long time and be disease-free. I became a scientist and always wanted to work on nothing except the aging process. When I was ready to do a postdoc, I knew I had to get a job eventually, so I decided to learn some immunology, figuring that no one wanted to hire you if you were working on aging, but a lot of people would hire you if you wanted to work on immunity. So I started to look at age-associated changes in the immune system, and I worked on that for 10 or 15 years. The more and more I got into that, the more I became convinced that it was interesting stuff and a lot was being learned, part of it by my lab, but that the real payoff was going to come from looking at things that slowed the aging process. In 1996, Andrzej Wartki published the first paper showing that a single gene mutation could extend lifespan in mice by 35 or 40%. It had already been known for a long time that calorie restriction could do even better than that. So the basic goal of the lab since about that time has been to see how these genes slow aging, how the diets slow aging, and eventually to find drugs that also slowed aging. That's now the central focus of the lab, is common biochemical and endocrinological and physiological pathways that slow aging, regardless of whether the intervention is genetic or dietary or pharmacological. About 15% of the lab tries to cheat by using natural selection. That is, we have fibroblasts from long and short-lived birds and long and short-lived rodents and long and short-lived primates. And we often will do a survey study saying, hey, does this enzyme characteristically go up if you have a long-lived animal in any one of these independent clades? If the answer is yes, and we've sort of discovered half a dozen of those, then we can ask, well, okay, if you have a slow-aging mouse, is it because that enzyme has changed? We can use the comparative biology part of the lab as a way of getting leads on targets that drugs or genes might use in an individual species like mice to slow the aging process down. So that's a sort of an overview of why I abandoned immunology to devote myself full-time to biogerontology.
Yeah, that was a great overview, and I'm sure we'll go back through some of those topics you've already mentioned. But obviously one thing that's kind of been a core of your research has been working with mice. And so obviously a lot of people talk about how translatable is research in mice to humans, but I just thought firstly, why mice? What makes mice a good model? Why are they commonly used?
Oh, mice are the best, obviously, clearly and dramatically the best. You've probably noticed that worms and flies don't look like us at all, and a lot of our hormones they don't have, a lot of our organs they don't have, a lot of our neural processes they don't have. Mice have all of them, with the exception of the part of our brains that are good at doing calculations and getting Alzheimer's. Mice have everything we've got. And you could do it in humans. Humans would be even better, except that you'd have to live 400, 500 or 600 years to get an experiment done in humans. So mice are the exactly right sweet spot. They're so much like humans that a lot of the time, things that are true in mice are also true in humans and vice versa. I'm sort of tongue in cheek. I read all the worm literature, all the fly literature, and I pay attention to it because very often they will make discoveries that point attention to things one really ought to look at in mice. If you investigate 10 of those leads, then you'll find 2 or 3 things that are really important in mice. That's a very nice percentage. I'm much more impressed by 10 to 20 to 30% crossover hit rates than I am by 0%. So the work they've done is highly influential, even though most of it does not apply to humans or mice. Some of it does. Enough of it does.
And I guess mice are also incredibly important for the interventions testing program. So I thought maybe it'd be interesting to hear the kind of origin story of how this, firstly, what is the interventions testing program, but also how the idea initiated?
Sure. Huber Warner, who was at that time the director of the biology program at the National Aging Institute, about 20 years ago, put together a working group. Some people who worked on mice, some that worked on invertebrate models, a few that even worked on dogs. We got together in a motel somewhere in Bethesda and spent a day or two talking about the best way to systematically go about finding drugs that would slow the aging process. And there was a wide range of opinions. Not too surprisingly, the mice people thought we should work with mice, and the rat people thought we should work with rats, etc., etc. Huber, after hearing everyone's thoughts, agreed with me and with Arlen Richardson. Arlen and I were virtually unanimous in how we viewed the matter, and he asked me to put together a white paper for his personal use that would explain how one might design an interventions program that the Aging Institute could conceivably fund, whose main goal was to find drugs that slowed the aging process in mice. We understood that finding such drugs would have at least 3 important benefits. It would serve as a stalking horse for hopes that eventually one might find drugs that worked in people. I think that's a moderately long way off, but I think it's an important thing to remember, and for most people that's the most important thing. For me, and I think for Huber and probably Arlen as well, the other 2 points were at least as interesting. If we found some drugs that actually did slow aging and extend mouse lifespan, that would give us clues to molecular targets for how the aging rate was controlled. The most famous of these, I think, at this point is rapamycin, a drug that inhibits a famous enzyme, mTOR, the target of rapamycin. Once the ITP had shown that rapamycin would extend mouse lifespan, this makes it much more exciting to look at what TOR does, how TOR is involved in all sorts of age-associated diseases, whether early or late life changes in TOR might have profound impacts on aging. Similarly, every time the ITP discovers a new drug, and we have at least 4 with big effects at this stage, and a small number, an equal number with small-ish effects. Whenever we discover a drug like that, it points molecular and cell biologists at targets that they ought to look at, because those apparently are targets whose manipulation can modulate the aging rate. The third benefit of the ITP, I think, is that it generates a lot of popular excitement and change in the zeitgeist, the popular opinion of aging research. One of the most important problems faced by people who are doing serious aging research is that most people think of it as a crackpot science, as a field filled with charlatans. People are going to make a buck by selling you stuff that doesn't work because the FDA has special rules to help them sell stuff that doesn't work without having to prove that it works. And if you tell people you're looking at anti-aging drugs, they will look at you as though you were crazy. You are a snake oil salesman. You should please find something useful to do with your life. So the third benefit, I think, of the ITP is to try to put that stigma behind us. If there is a growing group of respected publications in decent journals, replicated by multiple laboratories, saying, this drug extends lifespan, and it slows cancer, and it slows Alzheimer's, and it slows collagen aging and eye aging, et cetera, et cetera. Once that begins to become part of the public knowledge, part of the conversation, then if you send in a request to your congressman, I think, you should put more money into aging research. You're not laughed off of the block. That's going to be a long, slow, frustratingly slow process, but it doesn't start until there's a substantial body of good, solid, reproducible, peer-reviewed science showing that, by gosh, drugs do slow the aging process. I think those three things are the sort of main main benefits that the ITP has had over the years. Anyway, to get back to your question, once the white paper came out, the next step, the Aging Institute puts together a blue ribbon panel to write an RFA. No extramural scientists participate in this. It's just their staff members. Then there's a request for applications. Anybody in the world can apply. 3 people got good scores. My lab at Michigan, Randy Strong's at the University of Texas, and Dave Harrison's at the Jackson Laboratory. So the 3 of us got the first 3 grants. And it was important, I think, from the very start that there be 3 separate independently funded laboratories. We take a very unusual approach to science. Most people, when they get a grant, they are allowed to use that grant to do what they think is best for the science. The ITP is much more regimented. We have come up with a standard operating protocol. All 3 sites do that. We get the same food from the same kitchen at the same time. We use the same bedding. We use the same kind of mice. The data are pooled. The major benefit of this is that if a drug works at all 3 places, then we can say, this probably does work, and it may well work in your place as well. If it works at 2 of the 3 places, then we publish that, of course, but we say, this is worth a lot of thought. This may be a good working drug, but it's still a little uncertain. We need more data to see how robust it is, how strong the effect is. In the past, before the ITP, the world literature was filled up with single studies in which one lab did it with one kind of mice, usually, upsettingly, the black 6 mice, and would publish that, and then no one would ever hear from that drug or that study again. It takes, once a study is published, 3 or 4 years to replicate it, and a lot of money, and very few people want to spend lots of money and 4 years of their time trying to get the, oh yes, they were right paper, the replication paper. So having simultaneous replication at all 3 sites is a major benefit. The other major element that was insisted upon in the standard operating procedure of the ITP was we'd never, ever, ever use inbred mice. No black 6 ever. We use genetically heterogeneous mice. Any 2 mice in our program share half of their genes, like your brother or sister shares half of your genes. All of our mice are brothers and sisters in that respect. Because they're genetically heterogeneous, we will not trick ourselves into missing a drug that usually works but not on black 6, or in wasting our time on a drug that only works on black 6. If the drug works on our genetically heterogeneous mice, it's not a 100% chance, but a much better chance that it will prove to be robust over a wide range of different kinds of mice. So those are the central features of the ITP design. Immediate replication at 3 sites, the use of genetically heterogeneous mice, and community participation. We don't make up what drugs to test. Anyone in the world could submit 1 or 2 or 10 ideas. There's a February deadline each year. And then we have a committee system that looks over those ideas and picks 5 or 6 or 7 a year. We test them in a short-term pilot to see if they'll kill the mice. If the mice look great after we give them the drug for 2 months, then we officially accept it into our longevity programs. And this way, anyone in the world who has a really good idea about a drug, either a drug their lab works on or a process, an enzyme that they think is important, they don't have $100,000 to test it in a lifespan experiment, but we do. And so we can piggyback on their good ideas and incorporate them into the design of the ITP.
Exactly, that was a really great explanation to the ITP. And as you say, reproducibility is a huge problem in science. And it's great to see that you do it at 3 sites, as well as having these heterogeneous mice, and as well as doing it in both males and females, which is also really important, given that, as we'll probably get to, there are some sex-specific differences in your findings.
Oh, yeah, absolutely.
And so I guess, so my next question is just, what are the most surprising findings that you've seen in the study so far?
Well, the most important finding, which I don't think is currently surprising, is that you can slow aging with drugs. I mean, by now, those of us who pay attention to this field know that to be the case, but it was not known to be the case when we began this. It was known by those of us who were paying attention that you could slow aging with single G mutants, but no one wants to change their DNA, and we wouldn't know how to do it anyway. And if you're willing to lose 40% of your body weight, maybe you'd live longer, but no one is going to do that either. So the real translational pathway to human preventive medicine is drugs that you can take with a glass of water every morning. So that's the most important of the ITB findings, that's not science fiction. That actually works. Then amongst the more specific details, I found the most exciting original finding was in the 2009 paper, the Rapamycin paper in Nature. I would have bet, and I would have been incorrect, that for a drug to extend lifespan, you have to start it in young adult life. The earlier you start, the more chance you have to slow the aging process. And if you started it after the animals were already kind of old, and they already had some, pre-cancer, and their joints are a little rickety, and they had some endocrinological loss of beta cells, that it wouldn't work anymore. And that was wrong, because rapamycin started as late as 20 months of age. That's in human equivalent years. It's sort of in the mid-50s, somewhere along those lines. If you start rapamycin as late as 20 months of age, you still get a full lifespan benefit. I think that's really interesting. I don't understand it exactly. I mean, what it means, obviously, is that there are some bad things that can happen to you after the age of 20 months that rapamycin blocks, and that if you block them, lifespan is extended just as dramatically as if you had started the rapamycin in young adult life. We've now had data from 2 other drugs which are more or less supportive of that notion. I imagine we'll get to these drugs later, but acarbose, if started in midlife, gives half the effect that it does if you started in youth. And the other one, 17-alpha-estradiol, if started in midlife, gives an effect that's very similar to the same effect you would get if you started it in youth. So as a general rule, the notion that you don't have to wait, that is, you don't have to start with teenagers. You can start with people at the sort of level of seniority you'd expect of an associate professor or a tenured professor and still get a lifespan effect. I found that to be substantially surprising. And the third point I'll offer in response to your question, we have 2 drugs now, acarbose and canagliflozin, which probably work by blunting not the average amount of glucose in your blood, but the peak daily level of glucose in your blood. Neither of these in mice or people, well, at least in mice, seems to modify the average glucose level in your blood, but it sure does move down the daily high level, the peak level. And both of these drugs extend lifespan, and they both do it in males dramatically. One of them, acarbose, also works in females a little bit. Now that's a surprise. I would not have guessed that you could dramatically increase longevity by modifying the peak glucose hit. There's something in the body, one can guess it's in the brain, but we don't really know, that is paying attention to, oh my God, there goes the glucose again. Oh my God, it happened again. And paying attention to the very highest levels of glucose and using that to slow aging or do normal aging. I don't know how that works. Most of our mice are dying of cancer. So whatever it's doing, it's slowing cancer. If I was a cancer biologist, I would want to pay a lot of attention. How is it that you can slow down breast cancer and lung cancer and blood cancers and a lot of other kinds of cancers, liver cancer that these mice get, by blunting the peak daily glucose level? I don't know, of course, if it's going to work in humans, but it certainly works in mice. So that's a nice big surprise. Any one of our drugs that works gives us a whole new way of thinking about the aging process. The last surprise that I'll mention to you is work that is not ITP work. I'm going to brag about work being done by 2 people in my laboratory, Gonzalo Garcia and Jina Li, whose last name is spelled L-I. They have been working for 2 or 3 years to ask what appears to be a very simple question. Are there shared mechanisms of slowed aging? That is, if you list all the things that are accomplished by one of these slow aging genes, like the Snell-Dwarf gene or the growth hormone receptor knockout gene, you make a long list of those. And then you make a long list of the things that calorie restriction does. And you make a long list of the things that rapamycin does or acarbose does. Is there anything on all of those lists? And the big surprise is that, yes, we have 13 things, 13 famous things that affect fat and macrophages and brain and liver and muscle, which are flipped by 4 different mutants, 4 different drugs, and the calorie restriction diet in young adult mice. So these aging rate indicators, I hope, will become the sort of next big thing in aging research, because if it proves to be the case that anything that is going to slow the aging process will have to trip all 9 of these aging rate indicators and all 9 of those tissues, that gives us a way to screen for anti-aging drugs, and it gives us big clues as to what kinds of cellular and molecular processes are intimately connected with delayed and decelerated aging and the diseases of aging as well. That's more novel, and it's on the shakier ground. We have published 6 papers, but I want to say a lot of it's confirmed by other labs, and there's still a lot of work to do.
Wow, yeah, you've just raised so many questions, and I'm sure we'll try and go through some of those points, or at least as many of them as we can throughout the rest of this interview. But just to start with rapamycin, as you were mentioning, as being one of the most surprising findings, that you can start later in life and still have a benefit. Yeah. I believe since then you've also tested different doses, and have you also now tested different time periods? Does it have to be continuous treatment, or can it be sort of intermittent?
Yeah, we published a paper a few years ago in which we tried starting late in life, that is at 20 months of age, with 3 different treatment schedules. In one of them, rapamycin was turned on at 20 months of age, and they got it till they died. In one of them, it was turned on for just 3 months, and then it was taken away, and they never saw it again. And one of them was a monthly vacillation. It would go on for a month, then off, then on for a month, then off, then on for a month. For males, all 3 schedules worked great. They worked just the same. For females, there was surprisingly a big difference. Turning it on and leaving it on was great for females, as it always is. Giving it just for 3 months didn't do a darn thing for them. It's as though they'd never seen it before. The on-off thing was somewhere in the middle for the females. It was better than nothing, but it wasn't as good as leaving it on. We don't have any idea really why there was a sexual dimorphism in their response to these schedules.
Okay. And so, as you sort of mentioned as well earlier with rapamycin, it targets mTOR, which, it's involved in many processes within a cell, but one that it sort of promotes is translation. And I think this now sort of somewhat links to some of the actual biology research done in your lab, where you've shown some sort of work in CAP-independent translation and some potential investigations to understand why inhibiting mTOR might be increasing the lifespan.
Yeah, so those are two important areas. One is how does mTOR work, and the other is what is CAP-independent translation. And they are consistent with one another, but they point in slightly different directions. So most messenger RNAs are translated when the ribosome gloms onto the cap all the way at the 5' end, and then it zooms ahead till it gets to the start site, and then it makes a protein. But there's a subset of mRNAs, and no one knows how big it is. That a couple dozen, maybe even a couple hundred mRNAs are special, and they are eligible for CAP-independent translation. Their 5' untranslated region has a batch of special modified adenosine residues. The adenosine has a methylation at the 6th position, so it's called 6-methyladenosine. If CAP-dependent translation is blocked, and by the way, rapamycin blocks it, then the ribosome can start instead by jumping to these 6-methyladenosine sites that recognizes them with the help of a batch of other proteins, and it can translate the protein anyway, even though regular old CAP-dependent translation has been blocked. One of the things that Gonzalo Garcia found was that the liver and muscle and kidney cells from the long-lived mutant mice actually have lots and lots of CAP-independent translation. And then more recently, he found that mice on rapamycin have a lot of that. Mice on acarbose have a lot of that. And interestingly, 17-alpha-estradiol, which extends lifespan in males only, Gonzalo found that they have a lot of CAP-independent translation, but only in males. So it mimicked the pattern of results you see when lifespan is your endpoint. So of course, this doesn't prove that CAP-independent translation is a critical element of the slow aging process, but it is certainly consistent with that notion. And it may well be that the special set of proteins which are eligible for cap-independent translation are elevated in most or all slow-aging mice. We would love to know, is that true? So far, all of our data are consistent with that notion. And we'd also like to know, what are those proteins and what do they do? We have a clue about one of them, and now this is working on work that Jin Li has published. Special protein, an enzyme made by the liver called GPLD1. What it does is there are special proteins on the surface of most cells that are tied to the plasma membrane of the cell by a special sugar phosphate anchor, which involves phosphatidylinositol. So GPLD1 cleaves that anchor. And in so doing, it can throw a lot of proteins that used to be attached to cells, it throws them into the plasma. And we don't know what that does to the cells. We don't know what those proteins, the whole thing is mysterious. But there was a paper published a couple of years ago by another lab with two really fascinating findings. One thing they found is that if you exercise, GPLD1 goes up. And exercise, everybody knows GPLD1 is good for you. And then they found that if you overexpress GPLD1 in mice, your brain got good. That is, cognition was good, and also a protein called BDNF, brain-derived neurotrophic factor, that maintains brain cells in a stress-resistant formation, that went up too. So that was a clue that maybe the way exercise was good for you was it turned on GPLD1 and that did good things for your brain. Having read that paper, what Jina did was look at all of our slow-aging mice. And it turns out that GPLD1 is up in the liver, the fat, and the plasma of all 4 of our mutant mice, all 4 of our drug-treated mice, and calorically restricted mice. And for the drugs that are sex-specific, in some tissues, it's sex-specific for GPLD1. So the reason I got into this is it turns out, and Jina discovered this as well, that GPLD1 is a cap-independent protein. So
okay.
That's the link. We don't know exactly why cap-independent proteins are overexpressed in so many tissues of these long-lived mice, but Jina's work that I've just mentioned gives you a clue. One of those cap-independent translated proteins, GPLD1, has the ability to improve cognition, to improve brain cell turnover, and also to improve brain cell stress resistance. So that gives us, and we hope other labs, lots and lots of leverage for starting to look at that in molecular detail. GPLD1 doesn't actually get into the brain, and how it is that GPLD1 in the plasma causes the brain to do good stuff is not known. Other labs focused on GPLD1 are going to have to dig into that, but we don't know the answer.
Yeah, that's super interesting. And like, I have a couple of questions to sort of somewhat follow up with that. Firstly, I guess now that you have some more insight into mechanism, does that therefore lead to more potential compounds that could either activate, inhibit, like alternatively to rapamycin? And then also with this like cap-independent translation, is there any link as well with like immunity, and that like viruses sometimes, if they're trying to attack cells, they sort of exploit the cap-dependent? I mean, I might be misremembering my biochemistry, but could there be any link as well with enhancements of like cap-independent translation and having better immune response in the cell?
That's a good question. I don't know the answer. I think the people who are interested in immune function should really look into that. If maybe they've done it, I don't know the literature at all well, but if it turned out to be, as you were speculating, if it turned out to be the case that some pathogens, some pathogenic viruses turned off cap-dependent translation in multiple cells, one can imagine that preservation of cap-independent translation might be a cellular defense mechanism against infection. That would be cool if it was true. It may be true.
And yeah, and so have you also from this work found any other ways of maybe increasing the activity of GPLD1? Or I guess like generally it's more common to find inhibitors of proteins than activators.
Well, this is a great idea, and that's one of the things we're doing now. That is, we are screening a chemical compound library in vitro to say, do they turn on GPLD1? If we can find chemicals that are like FDA-approved drugs, which put onto cells in culture, turn on GPLD1, or even better, turn on GPLD1 and other cap-independent translated proteins, those chemicals will automatically get a place in the next set of ITP applications. So far, all of the drugs that all the 4 drugs that we have tested anyway, that are anti-aging drugs in the ITP, they do turn on GPLD1 and they do turn on CAP-independent translation. Now, the other way around is what we're going to do with our screening approach. We're going to screen for drugs that turn on CAP-independent translation. And if we find some, I hope we will, then we'll make an argument that we really ought to test those for lifespan.
Exactly. And I guess maybe this is another thing that could be looked into, is whether or not there's any sort of genetic variants, either in different species or within like centenarians, that are like in these CAP-independent processes. Just sort of speculating.
Yeah, I think centenarians are not the way to go, because they're so weird in so many ways. For instance, they're almost all going to die in the next year or two, and they've all had very extensive. They're all taking drugs of one sort or another. So I don't think that's the best way to look at it. But in the human population, there are good ways to look at it. Get yourself a batch of 65-year-olds and rank them into who is the healthiest. If you can find 10% of 65-year-olds who have great eyesight, great hearing, excellent reflex speed, perfect cognition, they can still walk for 5 miles without getting out of breath, and when you give them a flu shot, they produce tons of good anti-flu antibodies. Let's call that 10% the elite 65-year-olds. It'd be really nice to know whether they have a lot of CAP-independent translated proteins in their skin or blood. And it would be really cool to know whether they have a lot of GPLD1 in their blood. I would love to do that experiment.
Yeah, for sure.
Also, I guess a lot of humans that are taking drugs like, I don't know, metformin and canagliflozin, and see if the drugs increase GPLD1 in their blood and increase CAP-independent translated proteins in their blood. In fact, screening healthy volunteers who are taking any one of the 20 or 30 most commonly prescribed drugs you might stumble across a drug that did indeed cause that same set of aging rate indicators to increase in healthy young adults or in healthy middle-aged adults. I think that'll be the most straightforward and most relevant way of starting to ask these questions in human populations.
Yeah. And since you brought it up, I know metformin was tested on its own and didn't show a lifespan extension in the ITP study. Right. But then when it was combined with rapamycin, you saw an enhanced effect. No, is that true? And has it been like followed up?
Not exactly. It's a little bit complicated. When we added rapamycin and metformin together in females, we did not see a statistically significant increase in females. Rapamycin is great. Adding metformin to it, it's still great. It does a little better, 2% or something like that, but it could just be a chance effect. When you added metformin to rapamycin in males, you saw a bigger effect, but it still didn't quite reach statistical significance. The p-value was 0.06, achingly close to the standard conventional p-value. That's a troubled experiment because we didn't have a simultaneous rapamycin-only group. We were comparing rapamycin plus metformin to 2 previous our own historical data sets with rapamycin, which is always a little risky. So my own view is that it's up in the air, that if you add metformin to rapamycin with a large enough number of mice and maybe over 3 or 4 different doses, you might find an additive effect. However, we now have an additive effect. It's rapamycin plus acarbose. This was just accepted for publication a month or 2 ago. When you add rapamycin to acarbose, remember, acarbose is a drug that has a great effect in males, almost as good as rapamycin, actually. In females, it is statistically significant. It always works, but it's much, much smaller. In the males, it might give a 20% increase. In the females, a 5% increase. Always there, but always smaller. So in any case, when we added rapamycin plus acarbose in the females, it was just as good, but no better than rapamycin by itself. However, when you added acarbose to rapamycin in the males, you got the biggest percentage increase we'd ever seen. In the males, we're getting now a 29% increase in median lifespan if you combine rapamycin with acarbose. So that's 2 milestones. The first milestone is the one I just mentioned. It's the largest percent increase we've ever seen in the ITP. The other milestone is the first time we've ever seen a pair of drugs that used together gives you a greater percentage increase than either drug used by itself, and to a significant extent. So that's important, because you'd hope that using two drugs that have different targets and different mechanisms, you might get a bigger effect than either drug used alone. That's sort of expected. That's okay. But nonetheless, having data to prove it is a step forward.
Exactly. And I mean, since you've mentioned acarbose and also previously canagliflozin, I'm not sure if I'm saying it correctly, but they're all I guess effective sort of diabetics drugs to lower glucose levels. And so do they all, I mean, I know with metformin it's still not entirely clear the mechanism of action, but with canagliflozin it targets SLGT2 or SLGT1. And I guess I was just wondering, in those cases then, would it work to use those in combination as well?
Well, there's a batch of things to say about that. It's not known how canagliflozin works, either in mice or in people.
Okay.
Our starting hypothesis, which is reasonable, is that it's working by blocking in the kidney SGLT2, and in so doing, it lowers blood glucose. Because when blood glucose goes up, when the mice eat a few donuts or something, blood glucose goes up, and it is lost in the kidney if the reabsorption pumps are blocked by canagliflozin. That's a very plausible idea. However, SGLT2 inhibitors and SGLT1 inhibitors, they have targets in lots of other organs. And it could well be that the good things that canagliflozin are doing are either partly related to glucose and partly related to some other mechanism, or it could be that the glucose is just really a tiny part of the story and that some other target tissue really counts. In humans, clinical work, it's a drug that has been in clinical practice for diabetes only for a fairly short time, 5 or 6 or 7 years, something like that. But people have begun to notice that humans taking canagliflozin or other members of the family, other relatives that block SGLT1 or 2 or both, they tend not to get quite so much cancer, and they tend to get not so many heart attacks and strokes and stuff. So whether it has an effect of that sort in non-diabetics, I think is an interesting proposition. My hunch is that it does, but again, more data will be needed. It's a whole family of drugs whose targets and involvement in pathology are not yet fully understood. And so we don't really fully understand why it is extending lifespan in mice. I would love to see someone suggest to the ITP for next year 1 or 2 molecules that block SGLT2 only, and 1 or 2 that block SGLT1 only, and maybe 1 or 2 that block SGLT2 but only in the heart. There are such drugs. Because the drug companies, of course, are fascinated with this whole family of compounds, and they're making lots and lots and lots of compounds that are tissue-specific or target-specific. I would love to see half a dozen of them tested by the ITP, and that would help us discriminate the original hypothesis, it's blood glucose that's doing it, from other hypotheses which are not yet very well tested.
Yeah. And so as you said, these glucose transporters are obviously not just necessarily present in the intestines, but present in different tissues as well.
Yes.
And so clearly it's an important membrane protein.
Yes.
But have there been any sort of mouse studies, either knockout mouse lines without these transporters or knockdown studies that could also help to validate some of the intervention testings?
I think your question is a good one, but let me say 2 things about it. It has to be done by laboratories that are focused intensely, obsessively on SGLT2. So, you don't want an amateur lab, a lab that just does aging research, to be competing with laboratories which have 10 brilliant people working on SGLT2 inhibitors, because they're going to win and they know what they're doing and they have all the tools. The other thing is that knockout mice and even knock-in mice often will lead you down the wrong path. Often knocking it out is lethal. Knocking it out in a single tissue may not work because the real effect may be on a different tissue. So the sort of default assumption, let's knock it out and see if we get living forever mice, is too simple, and it occasionally works, but much more often the results are subtler and involve tissue-to-tissue communication. To give you one example, I told you about this GPLD1 story where GPLD1 goes up in a lot of slow-aging mice, including mice that don't make a lot of growth hormone. So we figured, well, obviously the liver is not making GPLD1 because it's not getting growth hormone signals. So we did more or less what you were suggesting. We knocked out growth hormone receptor in the liver only, and we expected to see GPLD1 go up, and it didn't. That was a big surprise. It's clear that what's going on in these mice is that there's some tissue, we don't know which one it is yet, that sees growth hormone or not, and then sends a signal to the liver that says, turn on GPLD1. So it's at least two tissues involved in this, the liver, which makes the GPLD1, and some mystery tissue that actually responds to growth hormone. We have at least one example where we know what tissue it is. There's a lot of stuff that goes on in the fat of these slow-aging mice. For instance, uncoupling protein 1 is up in the fat. It helps keep you lean. It is important in thermogenesis. Well, it turns out that the growth hormone receptor in the fat doesn't make any difference. It's the growth hormone receptor in the muscle. The muscle sends a signal to the fat telling it, turn on UCP1. So that there we actually found out what tissue does it, muscle, what molecule goes to the fat, it's called irisin, and what it does in the fat, it turns on UCP1. We haven't tracked that down yet for the liver and what is causing the liver to make GPLD1. We're going to have to knock out growth hormone receptor in this tissue or that tissue or that tissue and see what we get.
So is that mainly how you've been investigating this communication between the different tissues by using tissue-specific knockouts, or like what sort of tools are you using?
I think that's a good way to do it. Now, I'm lucky enough to have a collaborator, a guy named John Kopchik, who made and gave us liver-specific growth hormone receptor knockout mice, fat-specific and muscle-specific mice. So we have them in the colony. And whenever we get something like this, we sort of automatically try it out on those three organ-specific knockouts. In the fat, it answered our question. It was the fat sending the muscle sending a signal to the fat. In the question of GPLD1, it didn't. It's another tissue, a mystery tissue which we don't know about yet. And if someone comes to the lab and really wants to ask that, he or she will have to make brain-specific knockouts, hypothalamic-specific knockouts, macrophage-specific knockouts, and see what they get. It's sort of a guessing game, unfortunately.
And speaking of the brain, I think with the canagliflozin, there was a preprint. I didn't quite get time to fully read it myself, but I think you were looking into microglia cells and astrocytes in the brain in response to treatments. So is there any connection between improving brain health following reduction in glucose?
So there are two different labs working on this. In my lab, Jin Lee has looked at the brain and looked at two specific markers, BDNF and double cortin, which is a marker of brain cell turnover and neuron production. And she has found that they are indeed increased, which is the right direction, the good direction, by a carbose, rapamycin, 17-alpha-estradiol in males, and canagliflozin in males. Now, she did not look at glial cells, but I have a former postdoc, now running her own lab, a woman named Mariana Satigersky. She's an assistant professor at Wayne State down the road in Detroit. Mariana is an expert on glial cells, and she has found that a carbose and 17-alpha-estradiol, when given to male mice, prevent age-associated changes in glial cell activation and inflammation. They don't do that in female mice, which is consistent with the fact that these two drugs have a big lifespan benefit in males only. So Mariana has just published a paper, maybe the one you were talking about, in which she evaluated glial cell inflammation and inflammatory markers in both, I think, hippocampus and hypothalamus. But she is the real expert on that topic, and I am not.
Cool. All right, fair enough. So given what we've sort of spoken about and some of the results you found, what is your current theory of aging, or do you have a theory of aging?
I think it's the wrong question. I think to make progress in aging research, one has to stop asking about what is the theory of aging and start asking a closely related question. What is the theory of slowed aging? So somebody who works in immune function all too often will say, oh yeah, it's due to too much immunity. You're going to have autoimmune disease. That causes aging. Someone who's into connective tissue will say, yeah, it's connective tissue. Someone who's into the brain will say, yep, the brain. You've got to change the brain to slow the aging process down. If you're into oxidation or somatic mutation or whatever you're into, whatever your lab does, you think that's the cause of aging. I don't think that's a productive way to think about it. I think the bad things that happen to all of us as we get old, whether we're a mouse or a person or a dog, they have multiple causes. Some of them have to do with tissue injury that can't be replaced, or oxidation damage, or mutations that cause cancer, or cross-linking in collagen, et cetera, et cetera, et cetera. There are dozens to hundreds of these things that are the cause of aging. I don't care. That's of no interest to me. What I want to know is what can slow it down. The really amazing thing, which was guessable as early as the 1930s, but is now more or less unignorable, is that you can slow them all down together. You can slow them all down with at least 4 drugs in mice, at least 4 mutations, 5 mutations if you count P10 in mice, and at least 2 diets, the famous calorie-restricted diet, but also methionine restriction and amino acid imbalance diet. So since you can slow down aging of cells that divide, cells that don't divide, extracellular tissues, feedback circuits, immune circuits, since the whole thing, all hundreds of age-sensitive changes, can be retarded in parallel, the only really interesting question is, how does that work? What is it that is slowing everything down in parallel? Sort of as a metaphor, you can imagine a grand ballroom with thousands of lights in the ceiling, and if you wanted the place to get darker, you could send someone up on a ladder and take out one bulb at a time, or you could go over to the wall and turn the rheostat, and all of the lights will go down at a time. So people who are working on aging in the immune system, they're doing one bulb at a time, the immune system bulb, or aging in connective tissue, or aging in the brain. I want to know what where the rheostat is, and how the rheostat is connected to so very many cellular and acellular and multicellular and extracellular age-associated changes. When I got into this area, at the beginning of my career, it wasn't obvious that that was a smart play. It could have been. Most people thought, they may still feel, that the whole thing is too complicated and that you can really only work on one system at a time. Now I think if you're paying attention over the last 30 years, it's become incontrovertibly clear that you can change the rate of aging in multiple tissues and multiple cell types, and it's easy. That is, we have at least 4 genes and 4 drugs and 2 diets that can do it. So that's where you ought to spend all of your attention. How is it that these interventions that slow aging and postpone so many different kinds of age-associated diseases, how do they work? How are they what causes them all to change together? And what causes all of the downstream things, all the diseases and discomforts of aging to be linked? Not a perfect link, it's not a hard steel bar, but to be coupled to the aging process. Nature figured it out. If you buy a big dog, a Mastiff, that dog will be healthy for 6 or 7 years and then get sick. If you buy a little tiny dog, like a West Highland White Terrier, that dog is going to be healthy for 11, 12, 13 years and then get sick. Nature does this over and over and over again, makes short-lived breeds of dogs and horses, and also can make long-lived species and long-lived dogs and horses. It's apparently not too tricky. So I'd like to learn how to do it.
Exactly. So, what are your thoughts then on the field at the moment, kind of as it is? And what are your thoughts on the different ways of the biological age measurement? So things like DNA methylation, and how would that compare to these other potential markers you've been talking about?
Well, I am what we call an ill-informed skeptic in the area of DNA methylation. That is, many of the papers that have been published make it look as though DNA methylation is a way of assessment of multiple age-associated changes in many different kinds of cells. And they may be right. They're very smart people. They're very good scientists. It would be so nice if they were right. However, over and over and over again, they reach conclusions that, from my ignorant point of view, are far too good to be true. One reason I think to be very cautious about this is to note that every year somebody says, oh, the one that looked great last year, nope, mine is better. Each year they get better and better and better. So it looked pretty good 4 years ago. Why is it now not look so good? Another reason to be cautious is I sort of believe it if someone said, I've got a way of looking at your muscle or liver cells, and it changes between the time you're 20 to 40 to 60 to 80. Sure, that's reasonable. However, they say, oh, by the way, it works in all the cell types. Really? Every cell type has its own methylation pattern. And then they say, oh, yeah, and by the way, it also works between birth and adulthood. And now I start to say, no, there's something horribly wrong here. And then they say, oh, forgot to mention, it works in tissue culture, this thing we call cellular aging, which has been overhyped since Hayflick introduced that silly idea. They say, oh, by the way, the DNA methylation thing works in cell culture too, and discriminates young from old cells. It's at that point that there are alarm bells ringing all over the place. I don't trust even good scientists if they're making claims that always seem to work for them. I'm being deliberately a little iconoclastic here. A lot of these people are friends of mine. They know a lot more about it than I do. I send them tissues all the time because I want their work to succeed. And many of them are, I think, quite skeptical, and appropriately so. But I did want to introduce a note of caution. It's not, I think, ready for prime time yet. At the Gordon Conference on the Systems Biology of Aging, I really enjoyed a poster given by someone. They found that there was a marker of biological age that you could get from human blood, DNA methylation marker. And they found that when women got pregnant, they got old. And until they delivered the baby, then they got young again. And then they found that if people needed surgery, they got old until the surgery was over and they were back home recovering, and then they got young again. And if they were grad students, they were taking a test, they got old. Until the test was over, if they got a good grade, they got young again. So it's obvious that whatever they were measuring, which they were calling over and over and over again a marker of biological aging, it wasn't remotely useful as a marker of biological aging. It's a marker of sort of loss of physiological homeostasis, a loss of biological equilibrium. That's interesting. It's really interesting that you could detect many different kinds of stresses, including aging, by modulation of DNA methylation patterns. I think that's really cool. But to refer to it as a marker of the biology of aging and how old the cell is and how old the person is strikes me as sort of, what's the word, drug dealers using their own stash or something like that. I think they need to be a little more critical.
Yeah, no, it's given me a lot to think about. Kind of on from this then, what sort of advice would you give to, I guess, like younger scientists in terms of working out like why they can have the most impact and how to, like, what is the best way to do science? Obviously, reproducibility is very important, and I mean, it's well demonstrated in the ITP study, but what sort of like generic advice could you give to younger researchers?
Younger researchers who are really, really interested in the biology of aging? Is that the question?
Sure. I mean, I guess, well...
Or do you ask generically? For younger researchers, the answer is, and this is not a genius level answer, you have to work 3 times harder than anybody else. And you've got to really be working on something you care a lot about. Picking a topic that you think will get you published in a system where you can get results fast, that's often a pathway that burns you out if you don't care what the answer is. I think it's important to pick a problem that's really, really exciting. And then the other thing is, I think it's, as a footnote, important for you not to believe your mentor. Your mentor is a famous scientist, whoever you are, and he or she is firmly committed, with very rare exceptions, to the fact that their ideas are cool and really good and the right answers. I think you need to step back over and over and over again and say, no, wait a minute, this might not be right. There may be another better way of looking at it. Many people don't do that. They decide early in their career that aging is due to DNA mutations, or aging is due to we get too many senescent cells or something. And then they do that for the rest of their lives, and they're not interested in arguments or data where someone says, you know, it's not actually. It's not due either to somatic mutations or to so-called senescent cells. they don't want to think about that. It would hurt them to think that the last 10 or 12 years of their lives have been focused on the wrong problem or an uninteresting problem. I think you need over and over again to do that very tough step of saying, maybe I need to rethink this. It's hard for people.
Yeah, I know for sure. And so to kind of wrap up then, what are the sort of main questions or main experiments that you would like to be seen conducted within the next 5 to 10 years, and what sort of outcomes, or like where would you like to see the field in the next 5 to 10 years?
Well, I think the main interesting question is the one I've mentioned to you. What is it that slows the aging process? In mammals in particular, although there are good hints from invertebrate systems and there are good hints from cross-species, comparative biogerontology. So from all of those areas, if you read the literature, you can easily come up with 10 or 12 or 15 good ideas that are not yet disproven. Some people say, NAD and nicotinamide metabolism, that's a critical thing. Others will say, actually, mitochondria are a critical thing. Others will say it's major changes in the fatty acid composition of membranes. Each of these ideas is pretty good, and the ones I listed are not disproven. There are disproven ideas, like the cell senescence idea, but many great ideas are not yet disproven, and I think grabbing a hold of one of those ideas and becoming an expert on it is one way to make progress. I personally think, as you've guessed from our entire conversation, that it's necessary to keep all of those ideas open, but to focus instead on the central focus. What is it that is involved in slowed aging, regardless of how that slowed process comes about? And the more drugs we have that slow aging, the more mutations we have that slow aging, the more leverage we'll have for starting first to look at a sort of a checkerboard. If here you have 15 things that slow the aging process and 25 things you've looked at, which ones light up right across the board? Once you have a batch of those, then you can take the next step. Can I find drugs, or can I find mutations that change those aging rate indicators, those potential rheostats in the metaphor that I was just using, and thereby slow the aging process down? I think that's the pathway. Of course, that's what I'm doing. So naturally, I think that's the right way to go. I imagine others have different ideas, but that would be my own personal answer to your question.
Oh, yeah, no, thank you for that. And I think that's a great way to end our conversation. I think we've covered a lot of topics, and it's really interesting to hear how everything sort of fits together and goes from early observations in the testing programs, then now doing this whole molecular understanding and trying to work out the mechanism, which obviously is also really interesting. So yeah, thank you for coming on the channel today. It's been a really interesting conversation.
Well, thank you for inviting me. It's been a pleasure. I look forward to seeing this and other episodes of your podcast, of your show.
Oh, thank you. So I hope you've learned something in this video. Thank you to my Patreon supporters, and thank you for listening.